Ladies and gentlemen, welcome back to the post-lunch session of MycoIndia World Fungus Day 2023 lectures series, actually. Now we have Dr. Sudhir from Agarkar Research Institute. He'll be talking about whole genome sequencing of plant pathogenic fungi. I request Dr. Romana Mirdi to introduce the speaker, Dr. Sudhir, to today's audience. Dr. Romana, please.
Thank you, sir. Dr. Sudhir is working as a scientist at DST Agarkar Research Institute, Pune. He has obtained his PhD from Banaras Hindu University in mycology and plant pathology. His academic pursuits encompass the fields of plant pathology, fungal biology, host pathogen interaction, and disease management. He also demonstrates a keen interest in the field of fungal biology, specifically focusing on topics such as fungicide resistance and related concerns, hemobiotropic and biotropic fungal plant diseases. He is presently engaged in research pertaining to investigating the dynamics of host pathogen interactions and disease resistance mechanism, as well as the selective breeding of wheat, mung bean, and soybean crops with a focus on enhancing resistance to both biotic and abiotic stress. With this brief introduction, I welcome you, sir, and now it's over to you. Thank you.
Thank you, Ramana, for briefly introducing me. So, I'll be talking today about genomic mysteries of pathogenic fungi, how to unravel them with some case studies using whole genome sequencing, and horizontal transfer of some few what we call is a effector for disease causing fungi. So some major plant pathogenic fungi and their host, they belong to Basidiomycetes, Ascomycetes, and they have very broad host range. And several plant pathogens are being sequenced, and their whole genome sequence are readily available. And I have listed very few, but they are major plant pathogens. For example, Puccinia graminis tritici and Zynoseptura tritici, they are major plant pathogen in wheat. And based on their life cycle, either they are biotrophs or hemibiotrophs or necrotrophs. And their genome features majorly linked to their pathogenicity are repeat rich gene clusters that encodes effector candidates. Some other plant pathogens includes Plumeria graminis tritici, then Magnaporthia oryzae, a plant pathogen that is havoc for rice and now been on wheat also, and then Fusarium graminarium and Fusarium solani and other pathogens. So basic of host pathogen interaction that mainly relates to the this disease triangle that includes pathogen, environment, and host. And when we go for any genome sequencing, and that is mostly we try to dig out why the plant pathogen, what is the major factor those cause the virulence for the plant pathogen, or aggressiveness of the plant pathogen, and adaptability of that plant pathogen to different geographical locations and different environments and different hosts. And on the host part, that is always resistance mechanism, its growth habit, and new variety that helps in that either allow pathogen to infect or not to infect. And of course, environment part is also very important and essential, and that helps in survival of the pathogen, adapt and evolve. So when we talk about plant pathogen interaction or host pathogen interaction, these two models are largely discussed. One is trench warfare scenario, and another is arm race model. This trench warfare scenario is just parallelly talked with this gene for gene model of the plant pathogen. This can occur in various kind of environment and in different natural ecosystem. And this local adaptation of the pathogen leads to the diversity of effectors, those present in the plant pathogen and host pathogen population that maintain over the evolutionary time. And that is how pathogen evolves, and several races are being present in the environment. Then this positively diversifying selection, what is called balanced selection, maintains multiple alleles in host as well as in pathogen over the evolutionary times, and these are a random occurrence during the evolution. Another is R-race model. This model is generally occurs in agricultural ecosystem, where new alleles of the host population can be introduced very rapidly. You can see as a blue line here, and which drives the recurrent fixation of alleles in the pathogen also. Here orange and yellow lines. So start of new season at any when the crop start growing in every new season, every crop, like for example rice, wheat or anything, will have a new resistant allele that is to be introduced, and that is how this helps in evolution of the pathogen. In that case, frequency of virulence allele here in orange is low, but over the time when this new variety spreads across the large geographical area, pathogens start evolving over the time, and the allele that is that can adapt to this new resistant allele increases, and pathogen population reaches to the fixation, and new race get fixed, and that become the resistant variety becomes susceptible one. So when we talk about fungal pathogen genomes, they are highly diverse. These genomes vary in total size, then repeat content and gene number. Also, due to evolution and their diverse nature of growing habit, these vary from few chromosomes to many and include accessory chromosomes in particular pathogens, and these accessory chromosomes have several genome compartments also. So what are the accessory chromosomes and how they get introduced into the pathogen? So chromosomes, accessory chromosomes are those chromosomes, those are not present in all isolates of some pathogenic species, and these accessory chromosomes are often encodes the host specificity. For example, in biotrophic fungi, the host specificity is more, and these biotrophic fungi are very specific for that particular crop. For example, if you talk about rust fungi, the Puccinia graminis triticale is very specific for the wheat only. And if we take another example, the Puccinia hordi is very specific for barley. So these accessory chromosomes, those are get into the plant pathogen during the meiosis of sexual cycle of the fungi, and these are compatible mating partners. These evolves. They find out during the sexual mating, and during meiotic cycles, these accessory chromosomes are get into the that particular pathogen. The many species of fungi have mixed reproductive cycles, and they have both sexual and asexual stages. And the underlying mechanism, those are responsible for chromosome instability, because this accessory chromosome in several pathogenic fungi, we have found that these accessory chromosomes are lost during mitotic cycle. but the underlying mechanism that is responsible for this accessory chromosome are not known. Possible cause of the chromosome loss during mitotic cycle maybe the errors during the segregation of the sister chromosomes. So, characteristic of genome compartments. When we talk about genome compartment, the genome compartments from core gene includes lower gene density, higher transposable element content, and higher rate of evolution during the epigenetic marks. If we take an example of this heterochromatin region of chromosome 7 in Zymoseptoria tritici, a pathogen of wheat, the H3K27ME enrichment is a very good example. This chromosome 7 has example characteristics of both core and accessory chromosomes. And right arm is enriched with heterochromatin associated with histone mark, and locus close to the accessory chromosome may involve in fusion of accessory chromosome to the core chromosome. Then there is how this accessory chromosome get into the plant pathogen. The typical example is hybridization between Plumeria graminis tritici and Plumeria graminis spermaspecialis cicalis. So these hybridization leads to new pathogen, Plumeria graminis spermaspecialis triticale, and this is this pathogen infect on cecal cereal, that is what we called is a artificial cereal. So this is a extraordinary example that leads to the adoption of new host that is mediated by combination of parental virulence factor and emergence of accessory chromosomes for the host specificity. So we will talk about a few case studies. That is a discovery of virulence gene toxe in wheat and barley pathogen Bipolaris serociniana. This toxe effector is a typical example of horizontal transfer of the effector molecule from three different pathogens. And this is simultaneously reported, this jump, horizontal transfer was simultaneously reported from Australia, USA, India, and Mexico during these 18 to 20. So this toxe was initially discovered as a effector molecule, and in Pyronophora tritici graminis, and this was the only and major effector molecule that can cause disease on the wheat. In 2008, the first horizontal transfer, or we can say a jump of effector molecule, was reported from Parastogonospora nodorum. And after that, after 15 years, that again jump was reported in Bipolaris serociniana, first in Australia, then in India, then in USA, and then in Mexico. So why these kind of jumps are reported? So these pathogens, Pyronophora tritici, Parastogonospora nodorum, and Bipolaris serociniana, those are found to be grown together as a leaf blight disease in several areas, several wheat growing areas, and probable transfer of this pathogenic effector might be during this co-evolution of pathogen. So this 14 kb region of the Bipolaris serociniana of this toxe molecule has been reported as a transposable element across the three pathogen, and has found to be a very important effector molecule. Another typical characteristics of this interaction is this interaction is known for a inverse gene for gene hypothesis module, as this talks recognizes a gene in wheat that is called TSN1, and these toxic carrying isolates of either pathogen can cause disease in wheat only in presence of TSN1 genes. Knockout of TSN1 gene results in non-pathogenicity or avirulence in the pathogen. So these kind of jump are reported during the analysis of whole genome sequencing. You can see a haplotype network analysis of the different isolates, those carrying toxin. So small portion of Pyrenophora tritici means you can say a tox A haplotype, what we identified was closely related to the tox A of the Pyrenophora tritici repentis. So this resulted in new era, that is nomenclature of haplotypes across the fungal species, because different haplotypes of this tox, like 24 haplotypes of this tox A effector was reported based on the single nucleotide polymorphism, and they were identified or they were noted in a different names. So based on their homology sequencing and homology, the tox A haplotypes were coded from tox A1 to tox A24, and this is the network of haplotypes where they are closely related or diverse or distant from each other. So another study is global genomic analysis of wheat powdery mildew reveals association of pathogenic spread during the historical human migration and trade. This study included a sample of around 172 Plumeria graminis tritici genomes. And genome analysis showed that the BGT emerged in Fertile Crescent, that is Central Asia, Central Middle East, during the wheat domestication and spread throughout the Eurasia. And colonization brought this pathogen to America, where it hybridized with unknown grass mildew species. These pathogenic germs are just because of seed, those were carried during the trades and during the human migration from one continent to another continent. Then the trade between USA and Eastern countries, these strain were brought from USA to Japan, and European strain traveled from Europe to China. So you can see the differences between strains or genetics distance between the strains, but they were traveled from one country or one continent to another continent, and these similarity between the strains or similarity between the pathogen was just able to identify using the whole genome sequencing. You can see the migration path of these Plumeria graminis strains from these Fertile Crescent to USA and then to Central Asia or East Asia. So these whole genome sequencing were based on paired end sequence, and then high-sec, Nova-sec techniques were used during sequencing of these more than 150 strains of Plumeria graminis tritici pathogens. So In our lab, we started sequencing of few plant pathogenic fungi, and we we started with the Cercospora species, and those were pathogenic on several vegetables and other things. So 4 of our genome sequence are now available in several databases like NCBI, and they were published in IMA genome announcement section. And upcoming genomes include Cercospora capsicii, pathogen of chili, then Cercospora physalidis and Cercospora abelmushi, pathogen on brinjal. And we are now in process of metagenome of Bipolaris serociniana. So these are during last 4 years, there are more than 20 genomes. Those are made available by different labs across countries in single pathogen Cercospora. Now, when we started working in 2017 or 16, the only 7 to 8 genomes were available in repository. Now there are 29. So that is how rapidly genome sequences are available. And 3 of our genomes are now identified as a reference genome. Those are haploid assemblies, and these genomes are scaffold level genomes. So based on genome sequencing, we were able to find out the distance matrix showing the whether they are close or how they close and distant from each other. You can see all the blue highlighted genomes of our reported from us or sequenced from us, and these are their closely links. But we found that these genomes are very closely linked and less diverse, and it's very tough for us to identify these Cercospora species and differentiate from each other. So we tried to distinguish or find out some orthologous clusters and proteins in Cercospora genomes, and you can see there are around 7,000 clusters. Those are common in different pathogens. So further, we tried to find out unique proteins to in order to find out the effector molecules among the Cercospora species. So there were around 112 single element or protein molecules. Those were found. So we are in process to screen out those protein molecules. Those are common in major Cercospora pathogens. For example, Cercospora kikuchi and Cercospora peticula. These are major pathogens on and having diverse host range across the vegetable and other cultivated crops. So we are trying to find out the new effectors or effectors. Those are necessary for the disease causing organism. So there is there are different resources available to get insights to the fungal genomes, and very particular to the plant pathogens, and one of them is fungal genome resources is MycoIndia, and they have one project called 1000 Fungal Genome Project. So they are sequencing at least 2 pathogens from each family, and anyone can participate into that. So these fungal genome project has different kind of resources available, and they are giving the better insights using several tools. And they have used database of effector molecules, unique proteins identified with them, and so on. So they have one big project. What I have seen is they have sequenced more than 1,000 different isolates of one of the wheat pathogen called Zymoceptoria tritici. And this one particular paper reports its coevolution and of Zymoceptoria tritici along with the origin of wheat in different regions of and different continents like Central Asia, Middle East, then Australia, then Europe, and South Asia. And this particular paper shows the use of whole genome sequencing to track the spread of one particular pathogen and adoption across the diverse, you can say, environment, including the temperature range, then humidity and different varieties and different wheat species related to each other. So where do you find or where you can publish the genomes of plant pathogenic fungi? There are four major journals, those are available. One is Phytofrontiers, that is published by APS, then Genome Biology and Evolution by Oxford University Press, then I am a Fungus, and Microbiology Resource Announcement. Out of these, I am a Fungus publishes multi-authored report without any article publishing charges, and the genomes are openly available for all. But other three subject to article processing charges. They have different article processing charges. This is general workflow for NGS to be that is followed very commonly. That is sample collection, then extraction of DNA and target DNA amplification and other things. I haven't described next generation sequencing or sequencing methodologies because that is again another topic. But yes, sequencing methods includes Illumina high-sec, then packed by nanopore sequencing, then pyrosequencing, and they cost differentially. In India, I have experienced that Illumina high-sec, nanopore sequencing, and paired end sequencing is easily available. But it costs around 60,000 to 1.5 lakh based on your size of sequences, size of genome, and again what kind of assembly you want to have, hybrid assembly or like that. So these are some beautiful GitHub resources where you can find out the free resources or free websites and other things, and several codes. Those you can use for analysis, data analysis of the genome sequencing and visualization of genomes in a better way. So this 1,000 genome project is developed this genome scale phylogeny of the fungi. This is just for information, and I have shared with you. Thank you for your kind attentions and giving me an opportunity for the talk during this World Fungus Day talk series. Thank you.
Oh, thank you. Thank you, Dr. Sudhir. That was a beautiful presentation. Thank you very much. I think it was very enlightening for especially for beginners and even for people with the mycology who have not gotten into whole genome sequencing stuff. Thank you very much, Dr. Sudhir. Now we'll take up three questions. First, Dr. Sunil Srivastava, please.
Yeah, excellent work, Dr. Sudhir. Congratulations.
Thank you, sir.
I'm just wondering if you people have taken parasexuality also into consideration you talked of the ancestral chromosomes,
Yes, sir. Yes, sir.
which is mainly doing rounds through sexual reproduction, right?
Yes, sir. Yes,
But
sir.
many of the plant pathogens actually are not having any perfect stage, and
Yes.
the sexual reproduction is not there. So I was just wondering, because parasexuality is a very, very important phenomenon in those fungi, and that
Yes.
results into variability and evolution.
Yes, sir. Yes, sir.
Ha-
Currently,
yes.
we are analyzing the genomes of Bipolaris sorokiniana in concurrence with this horizontal transfer of toxa. And our basic hypothesis is parasexuality is one of the reason that this horizontal transfer has occurred between these three different pathogens.
Okay, so you have looked into that parasexuality.
Yes, yes.
Yeah. Okay, I just wanted to know about that. Thank you.
Yes, thank you, sir.
Thank you, Dr. Sudhir Srivastava, for your questions and interacting here. Nice to see you here, sir.
Thank you, sir. Thank you so much.
Thank you. Yeah, now Mr. Arvind, please ask you.
Thank you, sir. So it was a very good presentation, sir. It was very insightful. May I know any online resources where I can start for genome annotation, which can help me out for the genome annotation, sir?
Oh, I will share this GitHub resources into the comment section.
Okay,
So that has this annotation also.
Okay, fine, sir. Thanks, sir.
Dr. Gunjan Sharma, please ask your question.
Yeah, a very wonderful presentation and very good work. I have just a query more than a question that are you guys also working on the functional validation of these effectors?
We are started because we are in process of identifying these effectors. We have did the functional analysis of this toxa, but in case of Sarcospora and other, because we have more than 100 plus protein molecules identified, we are going one by one.
Okay, so you're only focusing on the serial pathogens.
Currently, I have grant on serial. That's why I have focused on serial.
Okay.
But yes.
Yeah, very interesting work and really all the best. Thank you.
Thank you. Thank you.
Dr. P.N. Singh, please.
Yeah, sir. Dr. Navathe.
Yes, thank you. Hello, sir.
Yeah, good afternoon. How are you? Fine.
I'm fine,
Yeah.
fine, sir.
So I have very basic question this regarding Gymnoseptoria.
Yes, sir.
So Gymnoseptoria, it is a newly established genus, no? This, I think that 2012 or something.
Yes,
And
sir.
very few
Yes, sir.
species are there. I think the 7-8 species are reported.
Yes, sir. Yes, sir.
So this Gymnoseptoria, it is a copy of that Septoria tritici. It is a newly established of that Septoria tritici, or it is totally different.
No, it is evolved from Septoria tritici.
Yeah.
But its evolution is due to this intercontinental jump, because this pathogen is found from humid tropics to the cold.
Yeah.
From Australia to Central Asia to USA. So the evolution is mainly due to this intercontinental jump of this or trade of this wheat.
Okay.
And that was identified recently because of this sequencing of more than 1,000 isolates.
Yeah. So so means our in whole farm that existence of that Gymnoseptoria, it is there in this our wheat and other alternative crop like Citeria and other gramineaceae.
Yes. But yet it is not reported from India.
Oh, okay. Yet it is not reported.
No. We haven't yet reported Gymnoseptoria from India. We have reports from for Pyranophora tritici repentis, but we don't have culture. There is one report, but we don't have culture.
So that's why, because that now that Kraus, Braun, and that company, they have earlier that Palidocercospora, that Septoria. So earlier, whatever that host specific that on Ficus, it was there.
Yes.
So that is very interesting that this Gymnoseptoria, it is a matter of study that whatever this alternative host of this our apart from this wheat, barley, and other gramineaceae.
Yes.
That is very interesting. And okay, so right now we don't have Gymnoseptoria in our India.
No, we don't have. No,
I-
we don't.
I think that is a matter of isolation and collection of that material.
Yes.
And we
Yes.
can understand that whether
Yes,
it's
yes, yes.
available or not.
So we did diverse collection on
Yeah.
wheat blight, but majorly
Not
we
yet.
got Bipolaris serociniana only from wheat.
Okay. And one thing, just for curiosity, I'm asking this. You said that Blumeris, that graminis.
BGT.
Yeah. So it's a artificial, means it is a breed of that, which that Felleris or your Citeria. What is that? It's a new breed, means newly established genus. This artificial minor grain.
No, we have Blumeria graminis tritisae. Earlier it was only Brumeria graminis, but due to host specificity, it's been again named as Blumeria graminis tritisae.
Yeah. That's why I'm for interest. I'm asking that because minor grain and it is. Yeah.
Yes.
So very good presentation by your Navathe you have presented. So thanks a lot. Nice presentation.
Thank you, sir.
Yeah.
Thank you, Dr. Sudhir and Dr. P.N. Singh ji for the beautiful interaction here. If audience, more participants have questions or queries, you can interact with Dr. Sudhir in our MycoIndia WhatsApp group. Okay, thank you very much, Dr. Sudhir. Wonderful presentation. Once again, congratulations.
Thank you very much, sir.
Now we will move on or move towards beautiful world of mushrooms, especially the mushrooms from the beautiful state of Kerala. May I welcome our friend from Kerala, Dr. Arun Kumar TK. Dr. Arun, first of all, thank you very much for agreeing to give this presentation, and nice to see you on this platform. And
nice to see you, sir.
Yeah, it's the first time I'm seeing you. Thank you very much. I'll request Dr. Romana Mirde to introduce Dr. Arun Kumar TK to today's audience. Dr. Romana, please.
Okay, thank you.